phagocytosis and other function in killing the foreign microorganisms. So we're going to talk about complement system. Then function of T lymphocytes. In the T lymphocyte, we will talk about killer, helper, and regulatory T lymphocytes, and T lymphocyte interaction between antigen presenting cells, because T cells cannot recognize antigen without presented by some other cells. Then we will talk about active and passive immunity. Active means your body makes itself, and passive means somebody else makes, and then you use it. That is passive immunity. Some of the immune mechanism is against our own body, because immune cell get confused and start attacking our own cells. And that is called autoimmune disorder and autoimmune disease. So we're going to go over autoimmune disease too. Adaptive immunity is specific to invader. That means whatever antigen you expose to, your body will make immunity against that particular antigen. Let's see, if you are exposed to COVID virus, your body will make immunity against COVID. And that's why that is specific to COVID. That's why we call them adaptive or specific immunity, but specific immunity cannot be produced without the help of innate or natural immunity, okay? Innate immunity. So you will see several places there is bridge between the specific and non-specific immunity, yeah? So adaptive immunity is acquired ability to defend against a specific pathogen by prior exposure to those pathogens or antigens. Without exposure to antigen, there is no specific immunity. You must expose to it, either by disease or by artificial way, okay? Is mediated by production of a specific antibodies by T lymphocytes or by activation of the lymphocytes which attack the infected cell directly, okay? So here you go. There is the T cells and B cells. When they get exposure to antigens, you see, stimulates first we call them helper T cells. So T cells are several types, like cytotoxic T cell, the helper T cell, cytotoxic, the killer T cells. So when helper T cell get antigen, then helper T cell help B cell and other T cells to get active. So helper T cell now, after exposure to antigen, stimulates B cells. B cell give rise to plasma cell and memory cells. Plasma cell, if you see, are packed with rough endoplasmic reticulum. They secrete antibodies. And those antibodies are gamma globulins. You remember one kind of plasma protein is gamma globulins. That gamma globulin then defend against extracellular pathogen, means microorganism get into your body, they can get inside your cell, or they can live in your plasma or interstitial fluid and attack your cell. So these antibodies kills only the microorganism which is outside the cell in your blood plasma or in your interstitial tissue. So they defend against extracellular pathogen by binding to antigen and making them easier target for phagocytes and complement. That means when these plasma cells secrete antibodies, that antibodies will bind with a specific antigen where this helper T cell was activated to. And then once they bind, then it is like sugar coating of the bacteria. Then other phagocyte like neutrophil, macrophages come and eat them, engulf them. At the same time, there are certain protein molecules, which is called complement, they get active too. And then they help mast cell to produce more histamine. They help other phagocytes to kill the antigens. That's how they help. So basically, antibodies help sugar coating of the microorganisms so phagocyte can eat them, and they also help complement system, and then that activates another cascade of activation which ultimately help phagocytosis. Okay, on the other hand, these memory B cells keep those antibodies inside the cell. They do not release, but when they expose antigen, second exposure, you see, second exposure of the antigen, that stimulates these B cells, and then these B cells immediately, fast rate and in a lot of amount, they change into plasma cell and secrete antibodies. That's why when you expose to any foreign antigen first time, you make limited amount of antibodies to fight infection. But if you expose to same antigen second time or subsequent, your body is ready to make a lot, because this is the reason, because you have memory cell, you have memory of that particular antigen beforehand. And that's why, that means you are ready. So if somebody is first time attacking the country, what happened? Army are not ready. They can lose. But if they get ready for next time, what happens? They can defend. Same thing here. At the same time, once helper T cell are exposed to antigen, that T cell stimulates other T cells, and that is called active cytotoxic T cells. And these active cytotoxic T cells, then they do not produce antibodies. They directly attack the pathogens which are hiding inside the cell. That's why they defend against intracellular pathogen and similar any other abnormal cell like cancer cells. They kill them. At the same time, they also, another T cells becomes memory T cells, which keeps the memory of the same antigen, which was helper T cell exposed to. And next time when there is second exposure, T lymphocyte, a T cell easily becomes cytotoxic T cells and does its function. So summary is very simple. Remember at least this, and then I'm going to go in detail. This is summary. Then I will go in detail because specific immunity is slightly complex. So I want you guys to pay attention, okay? So let's see, first understand this thing. Once you expose to antigen first time, that antigen is exposed to your the phagocytes, macrophages. And those macrophages bind with antigen, and then they release a lot of chemical compounds which activates B cells, particularly helper, sorry, not B cell, T cell, particularly helper T cell. And then helper T cell help B cell and T cell to do their job, which I just explained here. This is the summary. For now, you have to know what does B cell do, what does plasma cell do, what is memory B cell do, what is memory T cell do, and what cytotoxic T cell do. And we will go. So before we come to this depth in knowledge of this function of B and T cell, we're going to talk about B and T cell, where they are formed and how they mature. Both B and T cells come from our bone marrow. Okay, B cell is called B because that was found first in the bursa of a bird. That's why we call B. But they are formed in the bone marrow, red bone marrow. T cells are also lymphocytes, so they are formed into bone marrow. B cell comes and mature in the bone marrow where it is formed. That's why we call them, or later they can mature into lymphoid and secondary lymph nodes. T cell, on the other hand, mature in a gland, and that gland is called thymus. Where is thymus in our body? That thymus organ is found in our body. Where is it?
In your neck?
Yeah, it is in the behind your thymus, behind your sternum. So if this is my hand, thymus, if you like turn around, here will be the thymus. And that's the location in early age. Our T lymphocyte from bone marrow migrate there and live there and mature. But after adulthood, like after teenage, after 25, 30 years of age, thymus is pretty much inactive. Then your T cells, like now, all of us, most of the T cells are maturing into lymph nodes. Okay. So that is the maturation. We're going to talk about them all. Okay. So let's see now what happened. Okay. So antigens, we talked about it last time. Antigens are molecules that elicit production of antibodies that specifically bind to those antigens. Antigens are usually very large molecules and they are always foreign. And antigens are foreign, that's why our body immune cells can distinguish antigen as non-self. And antibodies are usually made against, antibodies are usually made against foreign antigens, not against our own self antigens. Most of the antigens are large molecules and they have a number of antigenic determinant site. So let's see if my hand is an antigen, it will have 1, 2, 3, 4, 5, like several hundreds of antigenic sites where different kind of antibodies can bind. Okay. And once these antigens binds with antibodies, then they produce another kind of antibodies based on those antigen determinant sites. Lymphocytes specific immunity is a function of lymphocytes, and non-specific immunity is function of other leukocytes, and they are neutrophil, eosinophil, basophils. Okay. So lymphocytes are derived from stem cells in the bone marrow and are two types, T and B lymphocyte, which replace themselves by cell division, so are not depleted or diminished in numbers. They are dividing every day, and we are making new one. Lymphocyte produced by this process seed thymus, spleen, lymph nodes with self-replacing colonies. So they are produced, some of them remain in the thymus, and after like adulthood, there is no thymus active, so we mature our lymphocyte in the lymphoid organs like spleen, other lymph nodes. B lymphocytes. So which lymphocyte produce antibodies, by the way? T or B?
B.
B. B produce immunoglobulins, which is called antibodies, and antibodies circulate in your plasma. Plasma is liquid and it is called humor. That's why immunity by B cell is called humoral immunity, because they produce antibodies. T cell,
The memory T cells also produce antibodies too.
sorry?
The memory T cells also produce antibodies as well, right?
No, they don't. They don't.
They don't.
They don't. Only B cell produce antibodies. Memory T cells do not produce antibodies. They keep the memory of that T cell, and then they divide into the cytotoxic T cell when they expose to the antibodies, antigens.
Oh, okay.
T lymphocyte, on the other hand, T cells develop from lymphocyte that seed thymus, as I said earlier. They do not secrete antibodies. No, none of the T cell, whether it is helper T cell, cytotoxic T cells, killer T cells, they don't. Okay. What they do, they attack infected host cell if the microorganism is inside the cell. So let's see if we have a bacteria here, if we have a normal cell here, and then we have this red bacteria inside our cell, then this cell is infected. So this cytotoxic T cell will come and attack this cell. Okay. So they provide cell-mediated immunity because it is white blood cell, T lymphocyte, cytotoxic cells, which produce this immunity. That's why we call them immunity produced by cell, and that's why we call them cell-mediated immunity. Immunity produced by antibodies. That's why we call them humoral immunity. Okay, this is like a lot of T cells supply 65 to 85% of lymphocyte for blood and most of lymphocyte in germinal center of lymph nodes and spleen. These lymphocytes are migrating everywhere from one lymph node to another lymph node. They are going. So they can, like they are traveling so they can meet the antigens. Secondary lymphoid organs. So lymphoid organs, thymus and secondary lymphoid organs. Thymus is located below the thyroid gland, grows during childhood, and gradually regress, means become smaller after puberty, dry after puberty. So after puberty, like most of us, our thymus is pretty much ineffective. So our white blood cells, lymphocytes, T lymphocytes are maturing into lymph nodes or secondary lymphoid organs. They contain T cell that supply the tissues. T cell can be depleted by HIV, AIDS, or chemotherapy. Particularly HIV AIDS kill our helper T cells. These can only be replenished up to late childhood, and after that, repopulation is accomplished by production in secondary lymphoid organ. As I said, up to teenage, early childhood, your thymus is very active, and after that, same job is done by secondary lymphoid organs. So what are the secondary lymphoid organs in our body? They are lymph nodes, spleen, tonsils. Tonsils are the lymph nodes inside your throat. Pierce patches, they are the submucosal lymph nodes lying under the mucous membrane in your respiratory tract, genital tract, urinary tract, and all the lymphocytes migrate there, and then they mature. But one of the organs, secondary lymphoid organ, which is spleen, has another function. So spleen filters blood. They does lymphoid function as well as they filters your blood too. That means when your red blood cell get older, this is the location when red blood cell is killed and recycled. Okay, so let's talk about the B lymphocyte B cells now. When helper T cell exposed to antigen, then helper cell get active, and that active helper T cell help B cell to bind with bacterial antigen. You see, here is B lymphocyte, and there is pathogen rod-shaped bacteria. So how B cell and the pathogen interact? Pathogen surface has antigens, like their plasma membrane, cell wall of the bacteria has some protein, some glycoprotein, some lipopolysaccharide, and they are antigen. You see different kind. Some are this kind, some are this kind. So one bacteria have several kind of protein or glycoprotein or lipoprotein molecules. They are antigenic. And these specific antigen will bind with specific receptor on the B lymphocytes. And B lymphocyte receptors are also these B cell receptors are antibodies. Some of the receptors are antibodies, which can bind with the specific antigens. So once with the help of B cell, and there is like complex mechanism between how B cells, helper cell get activated which helps B cell to recognize this same antigen. Like, let's see if there is no B lymphocyte. Let's see if you have HIV, somebody has HIV virus infection. What HIV virus do? Which cell is infected by HIV? Helper T cell. Helper T cell. Yes. So let's see if helper T cell is not there or infected or less in number, then what happens? B cell cannot do this job. That means any bacteria can come and kill you. That's why opportunistic infection is common in HIV. Okay, so let's see, we have normal helper T cell, which helps B cell to recognize the surface antigen. Once they bind, what they do? B cell divide. So you see, once this B cell bind with this dot red antigen with their receptors, B cell divide into plasma cell and memory cell, plasma cell and memory cell. If you look into the memory cell and plasma cell structure, what difference you see inside the cell? Do you see any difference between plasma cells and memory cells?
Looks like the plasma cell has that like rough endoplasmic reticulum.
Yes. Plasma cells has a lot rough endoplasmic reticulum, whereas memory cell has less. Memory cell will keep the memory, genes, develop the genes, and memory for that particular, this dot gene. The dot antigen. Now, plasma cells will start making antibodies. You see, they are secreting Y-shaped antibodies here, immunoglobulins, whereas memory cells keeps the memory. So when B cell divide, some progeny become memory cells, like this one, and others become plasma cell that produce about 2,000 antibodies per second. And this antibodies has one important characteristic. That means this antibodies is specific to this antigen, this small red dot antigens. These antibodies will not do anything to other antigens. That's why it is called specific. Okay. So that is the active immunity. You are providing active immunity because your own B lymphocytes are making indirectly the antibodies. Okay. So binding of B cell to antigen also triggers cascade of reaction that activate complement protein. So here, this is the one function. At the same time, these binding of antigen and antibody activates the complement. So they also activates here complements. And then complements, what does complements do? Complement help the phagocytic cells complement the function of phagocytic cells to phagocytose microorganisms. Okay. So binding of B cell antigen also triggers cascade of reaction that activate complement protein. Complement proteins are proteins like enzymes in our plasma. When they get active, what they do? They make a complex of complement and they attach to the bacteria cell wall and makes hole on the bacteria cell wall and kill the bacteria. Okay, so complement protein can kill antigen bearing cells and promote phagocytosis. So who bears the antigens? The bacterial cell or microorganism. At the same time, the complement protein, which is not attached to the cell wall and they are free floating in the plasma, can activate opsonization and mast cell. So let's see opsonization. Opsonization is coating the bacteria with the antibodies. So complement system help the antigen to bind with antibodies. And once it bind, what happens? It is like tagged. Like when there is a tree cutting, what happens first? People go and tag the tree, which tree should be cut in the forest. So this when antibodies bind with the antigen that is tagged, and then phagocytes, macrophages, neutrophils, they come and start attacking that sugar-coated or buttered microorganism. And that is helped by complement system. Next function of complement system is activation of mast cell. Mast cell activation. What does mast cell do?
Brings in more.
Release what?
Histamine.
Histamine. Histamine release increase the diameter of blood vessel and increase blood flow in that area. So there is more neutrophil, more other phagocytic macrophage cells so that they can engulf microorganism. Okay. So these are the three functions of the complement activation. Now let's see the molecular structure of the antibodies. Antibodies is produced by plasma cells, okay? And they are immunoglobulins. They are part of gamma globulin plasma proteins. And all antibodies have the same basic structure, but they have certain region which are specific for particular antigen. And that's why we call them antibody specificity. There are 5 classes: IgG, IgA, IgM, IgD, and IgE. And these subclasses differ with respect to polypeptide. So these antibodies are the peptides. That means they are protein. Protein means chain of amino acids. So based on their polypeptide or chain of amino acid, they have different subclasses. In a moment, we're going to talk about those subclasses, okay? And where are they different? So polypeptide in the constant region of the heavy chain. So let's see. First, these are the 5 types: IgG, IgA, IgE, IgM, and IgD. And here are their functions. So let's go and see the structure. Immunoglobulin or antibodies are in the form of Y or Y shape. It has 2 heavy chain, you see, and then attached to 2 light chain. In the heavy chain and light chain, there is VH, CH, VL, CL. You see that? So VH is variable BH is variable heavy chain. CH is constant heavy chain. VL is variable light chain and CL is constant light chain. When they these antibodies by the electrophoresis method, and they can separate into like IgG, IgM, IgG, IgA, 5 types. And when you crystallize each individual, then they fragment into 2 fractions. The variable area is called FAB region, and constant region is FC region. So this FC, or constant fraction of the antibodies, are common in all those 5 types. Okay, so is a Y-shape, 2 long heavy chains are joined to 2 shorter light chains. When cleaved stalk by of Y, they crystallize and forms FC and FAB. FC is constant among different antibodies, constant among different antibodies, and arm of Y is antigen binding fragment. So this is specific for particular antigen. Okay. FAB contains a variable region that confers antibody specificity. Okay. So like this, we have 5 kind, IgA, IgM, and each kind can be specific for specific antigen. So based on what kind of amino acids are in the FC region and then variable region, there are 5 kind, but there is like hundreds of thousands of millions of kind based on the arrangement and combination of these amino acid. So this is a specific region, this is constant region. Now let's talk about the T cells. T cells, receptors protein. Do you remember on the B cell, there was receptor. That receptor was binding, acting as antibodies to bind with the antigen, okay? So T cell receptor protein, only protein antigens are recognized by T cells. So T cells recognize only protein antigen, and T cells will not recognize protein antigen directly. They don't recognize the antigen if they are floating in the plasma or in the tissue. T cell receptor cannot bind to free antigen. That's why T cells respond to foreign antigen when they are presented on the surface of antigen presenting cells. Okay, so what are the antigen presenting cells? Antigen presenting cells are macrophage, the dendritic cells, which we call Langerhans cells, or any other phagocytic cells. Okay, so they are antigen presenting cells which present antigens to the T cells. Then T cell recognize them with the help of again B cells, not B cells, helper T cells, sorry, helper T cells. Are you following me? So here, take home message is T cell cannot recognize antigens freely present in the plasma or body. Some cells must present that antigen to the T cell. So how they present? Let's see, phagocytes takes the antigen, then put inside the cell, make a phagosome, and then mix with lysosome enzyme, make phagolysosome, digest that bacteria, cleave the protein, cut them in smaller pieces, and then bring to the plasma membrane of the phagocyte cell, and then expose it on, attached to another kind of glycoprotein, which we call them MHC, major histocompatibility complex. Before I go to another slide, I want to draw here, so otherwise you will not get it. Okay, so let's see. Here is antigen presenting cell, phagocytes. Okay, and we have a microorganism which is outside and then it enters inside. When it enters inside, there is lysosome enzyme which kill them, and then it bring here a small piece, bring here, and then here is another glycoprotein which is the part of cell. Let's see, this is the glycoprotein, another color. And then the green thing. Let's bring the green one. What is green one? Sorry, not green one, blue one. What is this blue color?
The bacteria.
The bacteria, good. So their protein now, this protein, bacterial protein, is exposed on the red thing. So there is blue thing. So what is that red? This is called major histocompatibility complex, MHC. Now, when this phagocytic cell are presenting with the MHC on the surface, then T cell will recognize it. Okay, there will be better diagrams coming. Okay, just I'm priming you so you understand. Okay. Okay. So here you go. How dendritic cell present them. So here you can see the dendritic cells in the skin. Dendritic cells are Langerhans cells. Do you remember in the skin we talked about? This is dendritic cells. Another name is Langerhans cell because Mr. Langerhans found it. That's why we call them Langerhans cells. So now the antigens, which is the red thing here, is binding to, is taken by these phagocytic cells, which is antigen presenting cells. Then they process them, and through the histocompatibility complex, they bind and then bring to the lymph nodes. And in the lymph node, what happens? They are presenting to the
T cell.
The T cells.
And now T cell is activated. So activated T cell, then what they do? Again, they go here in the lymph node and then go and fight infection later, attack the infected cells. Okay. So fragments are associated with surface with histocompatibility antigens. These histocompatibility, you see this between this red and the plasma membrane, this part, the green part, that is a glycoprotein. And you know glycoprotein is the part of plasma membrane or cell membrane. So which are necessary to activate T cells to increase the chance of interacting with correct T cell. Dendritic cells migrate to secondary lymphoid organs where they secrete chemokines and attract T cells. So these also activate chemokines. That chemokines, this dendritic cell, when they bind with antigen, they secrete chemokines, and those chemokines then activate B cells, and then B cells produce another cytokines, and then they activate T cells and B cell again. It's a complex, but here you need help of helper T cell here and helper T cell here too. Okay, so activated T cell now. What does activated T cell do? Here you go. Co-receptors on helper and killer T cells. Okay, so helper T cell, it's plasma membrane, and it has CD4 co-receptor. This is just, CD4 is cluster of differentiation, kind of glycoprotein on the plasma membrane. And on the helper T cell, there is T cell receptor, which is another kind of antibodies. When helper T cell get this antigen from the antigen presenting cells, helper cell get active, and that helper cell then produce cytokines, which activates B cell as well as T cells. And then T cell can recognize the same antigen, this antigen, when it is presented by target cells. Target cells means cells which are infected with the cells like our epithelial cells. If it is COVID, then our alveolar cells. Do you know alveoli has wall, simple squamous cells? That cell will have this class 1 major histocompatibility molecule. And through this one, with the help of helper T cell, killer cell will recognize that antigen. And killer cell is helped by CD8 glycoprotein to recognize it. And CD8 is the part of killer cells. Okay, so this is like how T cells, both helper and killer, recognize the antigens when it is presented to them. So killer T cell get the help of CD8, and helper T cell get the help of CD4 co-receptors. So this is the same thing here. Killer or cytotoxic T cells carry CD8 cell surface marker, destroy body cell that possess foreign antigen. So let's see, when this killer cell get the antigen, now what happens? The killer T cell becomes cytotoxic, active T cells, and these cells destroy the cells that possess foreign antigens. So if any cells in the body, whether it is your alveolar cells or lung cell infected with COVID virus, or any abnormal cells which has cancer cells, which has cells inside are abnormal and become cancerous, then the cytotoxic cells comes, activated cytotoxic cells, and then they kill them by two ways mainly. They kill by cell-mediated destruction. So cytotoxic cells then produce perforin and granizymes. Perforin and granizymes both are the enzyme proteins. Are you following me? Perforin makes or perforate a hole on the plasma membrane of those infected cells or cancer cells and kill them. Whereas granizyme is an enzyme produced by cytotoxic cells which get inside the infected cells and degranulate their DNA. Victim cell DNA, cancer cells DNA. And if DNA of the abnormal cell is destroyed, what happens? Cell cannot survive. And that's how they kill it. So cytotoxic cells kill infected and abnormal cell by producing perforins and granizymes. Same question in the short answer question next exam, I will ask, what are write one chemical substance which is produced by cytotoxic cells which kills the infected cells? And this is the answer, either perforins or granizymes. Okay. So here you see the association of TB and other T cells. So once stem cells, which is in the bone marrow, get antigen, stay in the bone marrow, mature, become B cells, thymus or lymphoid, secondary lymphoid organ T cells. After activation, B cell becomes, some T cell become helper T cell. Helper T cell help B cell to make their progeny and clone and make memory cell, plasma cell, and to produce antibodies. Some of the cells become regulatory cells. And how we know they are regulatory? Because on their plasma membrane, there is cluster of differentiation glycoprotein peptide that is called CD24. What does it do? It inhibits the excessive function of B cells, so that B cell is not dividing in uncontrolled fashion. It controls. That's why there is negative. Whereas helper T cell, as you saw earlier, helper T cell help T lymphocyte to transform into cytotoxic or killer T cells or killer T lymphocytes. What does this killer lymphocytes do? Go to the infected cell directly, attach to it, produce granzyme, make hole on the cell, destroy it. Produce another enzyme, sorry, perforin that makes the hole, and granzyme, which get inside the cell and degranulate their DNA and kill it. Okay? That's why immunity produced by B cell is called humoral, because they are putting their antibodies in the humor, in the liquid. And cytotoxic or T cell function is cellular function. They attack the cell with the cell. That's why we call them cell-induced or cell-mediated immunity. Okay, so primary and secondary response. When you take a vaccine, or when you get exposed to microorganism first time, what happens? The production of B cell function and T cell function or antibodies are very slow in primary. You see here, first time, weeks, 1, 2, 3, 4, 5. So you have like, it takes like 2 weeks to reach, and then there is plateau phase. And then after 3 weeks, it is high. Like after 4 weeks, you have good immunity. But if you expose to same antigen again, you see within a week, the concentration of antibody is so high. And then it slowly goes down. But in primary, it goes down very fast. That's why most of the vaccine has like first dose and then booster dose or second dose. Okay. So on first exposure, which is called primary, and secondary, second exposure. On first exposure pathogen, there is latency of 5 to 10 days before specific antibodies are made, and that is called primary response, and secondary is secondary, and you can see the differences. Active and passive immunity. Active and passive immunity is when your body makes immunity, then we call it active immunity. When you get from other, then we call them passive immunity. So when you take vaccine, is that active immunity or passive immunity? When you take COVID vaccine?
Passive.
Passive.
The active.
It is active
Active.
because you take antigen. You are not taking antibody. You take antigen, then your white blood cell makes, your B cell and T cell make the immunity. So that is active immunity. It is artificial, not natural, but it is your active immunity. So active immunity dependent on secondary response to provide active immunity. Immunization induced primary response by inoculating people with pathogens whose virulence has been attenuated or destroyed, which is called vaccination. So vaccine is either bacteria or virus or their toxic products, which has been detoxified. Like in COVID vaccine, we take their RNA, and that RNA has virulence but weakened. So it induced like virus, but it does not cause all the symptoms. Sometimes we take whole bacteria, like polio vaccine. It's live, and we have weakened the virus and take the virus. And virus can induce, activate the white blood cells, but cannot cause disease. That is called vaccine. Sometimes we take only the protein particle, the toxins of that bacteria, and make them weak and detoxify and put into our body, like tetanus. Okay, and that activate our T and B cell. That's why that is active immunity. What is passive immunity? It is immunity, but not by you. So is immune protection produced by transfer of antibodies to a recipient from donor. That is passive. So if somebody has immunoglobulin now in the body against COVID, and you take their blood filter and take that antibodies and give into another patient, that patient will have immunity, but that will be passive immunity because his or her body is not making it. You are getting from someone else. So donor was actively immunized, whether it is from clinical infection, subclinical infection, or immunization. And person who
Dr.
receives this,
Shaw, is this like when a mother gives the antibodies to her baby?
yes, that is also passive because there is vertical transmission from the mother's immunoglobulin cross the placental barrier, get into fetus blood, and that is also passive. When you breastfeed, the immunoglobulin, particularly immunoglobulin A, pass from your milk to the baby, and that get into GI tract. From there, it covers the GI tract and it get into your blood, and that's why that is also passive. Okay.
Professor, would this also be when, like, let's say right now with COVID, they do blood transfusions with people that have already had it? Would that also be considered passive?
Yeah, so blood transfusion, when you take whole blood, you are getting immunoglobulins too, but there will be very low. Like if you take a little bit like 1 pint of blood from someone, there will be some immunoglobulin, but there will be low. That's why we take blood, donate blood, and then we filter and take only immunoglobulin from that blood or antibodies. Okay. So person who receives these ready-made antibodies is passively immunized, like we use these in a snake bite, rabies, tetanus, and hepatitis. And in this case, you do like in hepatitis, we do active immunity too. Like you vaccinate against hepatitis, you vaccinate against tetanus. So there is, like if you take tetanus toxin, that is active. If you take tetanus immunoglobulin, then that is passive. If you take hepatitis vaccine, then that is active. If you take hepatitis antiserum immunoglobulin, then that is hepatitis, the passive immunity. Are you following me? This is all I explained earlier. Here is difference between active and passive immunity. Go over it. So active immunity is it lasts longer, but you have to expose yourself to that. Passive is like short, and we give passive usually when you are infected with some disease and just to save life. Autoimmune disease are produced by failure of immune system to recognize and tolerate self-antigens. So like sometimes what happens, some of the cells, our body are hiding some of the cells. Like, do you know there is a thyroid gland? Thyroid gland has thyroid follicles, and inside the follicles are thyroglobulins, which binds with the thyroid hormone. And sometimes you get thyroid inflammation, and then some of the globulin, which is in the follicles, are coming outside and get into blood. All of a sudden, that own cells is like foreign because white blood cell has never seen them. And then again, B cell and T cell get active and start attacking their own thyroid gland, and that is called thyroiditis, Hashimoto thyroiditis. You see here, Hashimoto's thyroiditis. This is the scientist, Japanese scientist who found it. That's why we call this thyroiditis by his name. Similarly, other examples are like rheumatoid arthritis, lupus, psoriasis, rheumatic fever. You know, in psoriasis, in lupus, your body makes your own antibodies against your own DNA. In psoriasis, you have skin DNA against DNA. You make your own antibodies against your own cells, and that's why your cells are dying of mitosis very fast. So these are the autoimmune disorders. Autoimmune disorders are common in female than male. I don't know why. It is the way commoner in female. Okay, so that is all for today, guys. If you have any question,